No, we're Pascal. We're just waiting for one more person to show up. We got everything. Oh, we're ready.
Oh, really? Yeah. 5 minutes.
How late are you ready?
Half hour.
Because her grandma thought it's now. It's not ever half hour.
All right, now that we're here, I want to thank you all for coming. I'm Bob Mason. I'm professor in the Department of Integrative Biology. I'm David's thesis advisor. And I've been doing this so long, I often give a canned speech, not a canned, but it's it's one of those bittersweet things. It's like your children going to college or something. We get to interact with these guys for so long. They are kind of like your kids, except you don't get them cuddly and all the good stuff. We get them all grown up with all their manifestations of so, but not. So that's why I said it is bittersweet sending them out. David Hubert is almost one of these people that, as they say, he almost doesn't need introduction. One is because he is a Corvallis boy, born and raised, and so there's lots of people in the room here, family, friends, grad student colleagues, undergraduates. There's all kinds, which is a great tribute to David. And I know there are people online that couldn't come. And in some ways, I want to say I kind of struggled with how I was going to talk about this, but anyway, it's not my story to tell. But in many ways, David Hubert should not be here today. Let's just say his educational background is unique, and you can ask him about that. But so I don't want to be sort of man of mystery or something. It's not in any time under a bushel basket, but. In many ways, he most certainly did not take us for granted route here to OSU and to the grad school. I will say a couple of things. I know I have a good friend, Patty Petzel, who's over at LB at Lynn Benton Community College, and I've known Patty for probably 30-something years. And one day I think she even called me up. She said, Bob, we're sending a guy over there, and I think you should keep an eye on him, or like be aware of him, not keep an eye on him. Because my dealer used to say the other way. And I was like, that's pretty weird. Patty's never done that in all the time for any other person in my entire lifetime, knowing her. So it's like, better find out who this David guy is. And then we hooked him up with Lori to work in the freshman labs. But anyway, that was just to do his undergraduate work. So David's been here like 10 years, but you grad students don't have your sphincters clamped shut because it's not like a grad student. So he was an undergrad, he worked in our lab, and it was one of these that like, well, David, you should really think about going on doing graduate work. So he looked into those things, and then it became one of these things that's more and more common. Because I really like what I'm doing here. Do I have to go somewhere else? And of course, the answer is no. Increasingly, it's no. So of course, then he's he did stay, and he's done a really nice thesis for all of you grad students. Again, it's the kind of thesis that I would say is this fair, David. Some people do, and I did one myself. Do all that work, and it doesn't really come to fruition until the very end. Not like the thesis part, but it's like, I'm not sure I'm going to have a degree unless this thing hits pay dirt. It's not quite like that. But anyway, David's hit pay dirt and then some. So let's see, what else do I want to say? David, so some of those things, the more formal part. So I already figured David got his bachelor's degree here at OSU. And I guess you were what? Zoology or biology? Bio major. Then he did not do a master's. He went right through to a PhD. Again, very common for those of you not in this world. Common these days. While in graduate school, he was awarded a very prestigious National Science Foundation pre-doctoral fellowship. Again, all of us who know that's those are tough things to get. Very few of my students never got that. So that was great support there. He is also he is well known to undergrads, so he has spent a lot of time working on his teaching as well. He is really a gifted teacher, I would say, and I'm a pretty stern judge of that. So without further ado, and more of me rambling through here, David is going to talk about some of the work he's been doing for the last few years. And am I going to do the title, or are you going to do it? I guess all right. So David Hubert is going to talk about life in a rapidly changing environment, the thermal biology of the red-sided garter snake that lives in California. So David, take it away.
All right. Thank you for that introduction, Bob. And thank you all of you for showing up. I appreciate the support. We have a lot to cover, so we're going to get into it real quick. So first, I'm going to give you kind of the basic outline of what we're going to cover. We're going to go into the background, what I study, why I'm studying it, and then we'll break up the actual research I did into the 3 major seasons that these animals go through. And that'll make more sense after I get to the background, why we're doing it that way. Then we'll cover the conclusions and have time for questions at the end. So, story time. When I joined the Mason Lab, Bob told me that I could study anything I wanted, so long as it was garter snakes. Now, I didn't know a whole lot about garter snakes coming in. I had a lot of things I was interested in, and I wasn't sure kind of where I could meld these. So I started digging into the research on garter snakes. And it turns out there's a lot known about them. They're very, very well studied. This population we work with has had papers published consistently since about the 1960s. And so there's a lot of very specific things that are really well known in this population. So my challenge was to find something that wasn't very well known with them that I also found interesting enough to spend the next several years working on. So I started by looking at where they live. So the species we study is Thamnophis sertalis. It's the garter snake. And their range covers most of North America, right? All of these different colored areas are different places where they live. The specific subspecies we work with is Thamnophis sertalis parietalis, and they live in this yellow area here in the middle. And you'll notice that's still a really wide range, right? The southern end goes into northern Texas, and it goes all the way up into northern Canada, right? So spanning a whole lot of different areas and environments. The specific population we work with lives right here in a place called the Interlake region of Manitoba. And this population is really, really well known. They are not only the biggest aggregation of snakes in the world, but they're the biggest aggregation of terrestrial vertebrates as well. So they live in colonies of up to tens of thousands at these sites that we work with them at. They're so well known, in fact, you may have seen this driving around down the street. This is actually a depiction of, or in reference to, the population we work with, right? So they're a world-renowned population. Also, this area they live in is really well known for their very, very long, very, very cold winters, right? So the average winter temperature there is minus 16 degrees Celsius. So that's well below freezing. And these winters can last up to 8 months with constant snow coverage that whole time and below freezing temperatures. So very, very long, very cold winters. It's also notable that their spring transition between winter and summer is really, really variable, right? So you have average daily temperatures that go anywhere from 5 degrees to 20 degrees. It's a pretty big swing daily for averages. But you also see that within the same day, you can easily have snow falling with below freezing temperatures to like t-shirt weather in the same day, right? Where things fluctuate really rapidly there during this time period. So that kind of seems interesting in itself already, right? So I'm studying a snake, which is cold-blooded, and we'll talk about that in a second. And it's living in this environment that's very, very cold and very, very thermally dynamic, right? There's a lot of change going on there. So what do I have cold-blooded in quotation marks? Well, cold-blooded is kind of a bad term, right? They don't actually have cold blood. What we really mean is that they're ectotherms. And the distinction there is that they're dependent on the environment as their source of heat, right? They don't create it in their own body through metabolism like we do and other mammals do. They have to get it from the environment around them. And more specifically, the ones that we work with and most reptiles are going to be poikilotherms, where they're going to use behavior to regulate their body temperature. So and we'll talk a little more about how that behavior works in a minute. But the basic idea is they can actually create body temperatures that are higher than the environment around them using behavioral adaptations and things. Okay. So far, I found that they're cold-blooded animals that are dealing with extreme daily thermal variation and extreme seasonal thermal variation. So this seasonal thermal variation plays another role here, where it actually drives really distinct seasonal behavior that breaks into these 3 different phases that we're going to talk about. And these are going to be really important to understanding their basic biology. So first, it's a cycle. We got to start somewhere. We're going to start with spring, which is kind of their most well-studied season. Most of what we know about them comes from during this time period, this part of the year. And so in the spring, it lasts about a month. Usually it's the month of May. They all come out of their winter hibernation sites or their dens, and tens of thousands of them will be out all at the same time, and they engage in active courtship behavior. They do their mating during this time period. And they don't eat. That's pretty notable. They're just out there doing this behavior. And after they're done with that, they will migrate. For our population, it's about 20 kilometers to their summer feeding grounds. These are kind of marshy areas where they primarily feed on frogs. And they'll do all of their eating for their entire year during this time period, which goes as, if weather's bad, it can be as little as 10 weeks, but usually it's about 3 months each year. Notably, and we'll talk more about this later also, if females successfully mated during that spring mating season, they will actually carry their offspring to term during the summer feeding season at the same time period where they would have opportunities to feed. They actually give live birth. They don't lay eggs, so they'll carry those babies all the way until they're born. And that's pretty important. We'll talk more about that. And then finally, we have the biggest chunk of time is that those really long winter seasons. And they do something called brumation, which is very similar to hibernation, but it's something that cold-blooded animals are able to do. And this season, again, is characterized by below freezing temperatures, constant snow coverage for up to 8 months each year. And they go underground into their hibernacula, which are like limestone deposits with cracks in them. They can go down, and they go down to the water level. And they basically hang out in the dark with near freezing temperatures and no food for 8 months out of every year. All right, so very long periods there. So we've got these extreme daily thermal variation, extreme seasonal thermal variation, and this very distinct seasonally driven behavior. So we got a picture for me here. I wanted to back up and look at kind of the big picture and see what's going on. And so I wanted to see how climate change might play a role. And it turns out that when you look at that, this region of Canada actually has seen an incredible increase in warming compared to the global average. So if we look since about 1950, the global average increase in temperature has been about 0.6 degrees Celsius. For this region, it's been 1.7 for that same time period. So almost 3 times the average annual warming compared to the global average. And then if we break that down further for that same time period, we see up here in the spring where they're doing the really important courtship behavior and reproduction, it's increased actually by almost double that, so 3 degrees Celsius. So 5 times the global average for that time period. So yeah, there's a lot going on there where climate change, warming caused by climate change, could be playing a role in this organism and this population as well. So I found what I wanted to study, right? Thermal biology. There's so much going on with that. It covers all these different behavioral and physiological things and reproductive things. That's where I stuck, and that's where we're going to move forward talking about that. So like I said, we're going to break down the research by season that it's involved with, and we're just going to kind of march through starting with spring. So I was really focused on looking at the varied thermal conditions in spring. To me, that was kind of one of the more important aspects or more interesting. And so I kind of broke it down in some different experiments that I did and some ways to look at it. And we're going to go through those quickly here. So first, to kind of give a baseline, I wanted to establish their thermal preferences, kind of where they want to be during this time period, and then kind of what some behavioral limits are. So where they going to change behavior based on temperature. So the first question was to just really establish what were the internal body temperatures of snakes in the wild. What are their kind of preferences? To do this, I went to the den sites and I collected internal body temperatures of individuals. I stuck with males for most of the temperature stuff because males and females actually have a pretty big difference in their thermal biology, we think. So I stuck with him to kind of keep it consistent all the way throughout, just as a note when we go forward. So I went and got temperatures of active individuals in the field on 3 different days, on a cool day, a warm day, and then what was a hot day. And I used behavior as the indicator to pick individuals to get the body temperatures from. On the cool and warm day, there was normal activity occurring. So I picked individuals that were engaged in that activity, this courtship activity. On the hot day, there was not normal activity occurring. There was not very much activity at all. So I picked individuals that were doing a behavior that we call loafing, which basically means things are too hot, we can't do what we're supposed to do, we're gonna go retreat and try and cool down. And so I collected all that data on there, on these things. Then I wanted to move forward and look at kind of what body temperature is normal behavior altered, right? When does it get too hot to do normal activity that they need to do? So I mentioned that they can do behavioral thermal regulation, and it's really straightforward. This is essentially what it is. They can change what they're doing to alter their temperatures, right? And so if they're too cold, they can come out here and get on a rock in the sun and they can warm up. If they're too warm, they can retreat underground or go into the shade and cool down, right? And so they're really, really good at this, and they can do a lot to control what their body temperatures are by doing these different behaviors. So the first behavior I was interested in was courtship behavior. And I wanted to see basically, when was it too hot to court, right? When was it too hot to work, to do that activity? And so I set up courtship trials where I was able to put several males and a few females into an arena, prevent copulation. So I wasn't interested in post-copulatory behavior. It was actually that courtship behavior. And then once robust behavior was observed, I would then turn the temperature up until it got hot enough that they couldn't do it anymore, and then record those temperatures. The next thing I wanted to look at was what's called a voluntary maximum. So basically, when do temperatures get so hot that they want to get out of those conditions, when they want to escape that and cool down? And so to do this, I created a kind of high temperature thermal environment where they couldn't do normal behavioral thermal regulation, and I could watch for other behaviors that signified that they wanted to get out of that they were too hot and needed to leave. And so I was able to do that. And then further, those are the behavioral limits. I wanted to look at what their physiological limits were. So when does it become dangerous? When it's so hot that it becomes dangerous. So to do that, we do an assay that looks at loss of coordination. So basically, well, we're going to step through it here. I'm using a lizard as an example because it has legs. You'll see why in a second here. Legs are easier to see make it easier to see what's going on, but it's a squamate too. They all kind of react in the same basic way. So you'll take an animal and you'll apply a condition. In my case, it was either hot or cold temperatures. I was interested in both. And then you watch for a behavioral cue that signifies that something has occurred, something has changed. You test them for their righting response, which means you take them and put them on their backside, on their dorsal side. And then you watch to see if they're able to right themselves, turn themselves back over. If they can, then they have not reached their physiological limit. They've maintained their righting response. But if their righting response is lost, we consider that to be that physiological limit has been reached, and we record those temperatures and move forward. So I wanted to figure out what those were for both the hot and cold. And so I created, I used that same heat chamber and created a cold chamber, was able to rapidly change their body temperatures to either a hot temperature or a low temperature, and then tested them for righting response and recorded the values once they lost that righting response. So it was a lot really quick, a bunch of different experiments, but we're going to talk about the results here, and we're going to compare those kind of to how they stack up against other regions of the same genus or species and kind of historically. So this is the results from all of that on a nice little pretty rainbow graph. And basically what it gives you is this kind of range of behavior or temperature, where you've got that minimum physiological limitation here in the blue, and the red is the maximum physiological limitation. And basically, if they get past those temperatures, they're dead, or they're going to die. They can't escape them because they don't have coordination. So those are too high. So for the green area, we've got kind of the preferred temperatures. We actually found they were different between the warm and the cool day, but they were kind of mid to high 20s. How that stacks up against what's known for others of this genus, so really widespread, it's pretty much way lower. So we should expect it to be about 30 degrees Celsius. However, that's pretty normal because I looked at individuals who are engaged in courtship behavior, and it's been found that they actually will trade what kind of behavior they're doing. If they are wanting to do courtship, they'll favor that at the cost of behavioral thermal regulation. So it allows them to, focus on the things that are important, and they can still survive it. But what's interesting is that that cool day actually had significantly higher body temperatures with a lower range, a lower variation. And so what we're really seeing here is that as the environmental temperatures are further from ideal conditions, they have to invest more time and effort into that behavioral thermal regulation. And likely that's coming at a cost of other activities. And in the spring, it's going to be coming at a cost of the courtship activities. So it's not necessarily, there's definitely a strong trade-off here that they're going to have to exhibit for that. When we look at the behavioral maximum temperatures, our average is right around 35 degrees Celsius. And that's really what we would expect to see based on other members of that genus and kind of historic samples from the same population. When we look at the physiological limits, we see that the low was right around 1 degree and the high was right around 40. And again, those are kind of right in the middle of what we see for other closely related organisms there. So there's not a lot of difference in what we saw versus what we expected to see. So kind of the conclusions for this section, we see that their thermal thresholds are very tightly regulated and that their preference for behavioral, physiological limits were not very different compared to the historic or geographically distant relatives, which means they're likely mitigating these thermal changes through behavioral thermal regulation alone, right? They're not actually changing their physiology or their molecular work for that. They're changing their behavior to accommodate those differences. So that leads to a couple questions. So what happens when the change is so great that they can't overcome it with behavior anymore? Are they gonna, if there's a limit that's reached, how are they gonna overcome that in a very short period of time? And then additionally, how are they actually surviving rapid thermal change? Things that we see additionally with climate change or a lot during that spring season. So this last part kind of teases up for the very last part I did during the spring work, is looking at their acute thermal stress response. So really quick, what we did here is looked at their transcriptional response to acute thermal stress, but let's go over what I mean by this real quick, because I know not everybody in this room is familiar with transcription or what's going on with that. So to understand this, you have to understand this biological concept called the central dogma of molecular biology. I know it's a mouthful, but we're going to kind of simply go over it so you can get the basic idea of what I'm talking about, and then understand this experiment and another one down the line. So in your body, when you need something, our body is able to make proteins to do things, enzymes and various other things. And so in your cells, when you need something, a signal will generate saying, hey, we need a thing, right? And it will send it to your nucleus in your cell where your DNA is contained. And you can think of your DNA as being like a database of all the directions for all the things that it can make, all the different proteins. And so we'll go ahead and search up through this signal the exact directions to make the protein that you need. And then you basically print it out through a process called transcription, where you're taking that information encoded in DNA and turning it into a different molecule called RNA, making these things that we call transcripts. And you can think about that as being like the blueprints for that protein that you need. We'll do a process where we use those blueprints and the building blocks we have in our cells to create the protein called translation. And so this is the basic idea of what the central dogma is where we have stuff stored in DNA that's converted into RNA, and then that information is used to build proteins. So we go from there for those kind of 3 steps. For transcription, or the transcriptomics is the study of transcription, where I'm really just focused on that middleman. I'm focused on those transcripts. And how we use that is basically, we'll compare the numbers of copies of transcripts between different conditions and make determinations about what that means. And so in this case, you've got stacks of those spaceship directions for 2 different conditions. We would conclude that there's a bigger demand for spaceships in condition B than condition A. And this is a very simplified version of this, but this is the basic idea of what I'm going to talk about in this project and a different one down the way. This is what we're comparing between different treatments. So the question is, how do garter snakes cope with rapid thermal change when they can't use behavioral thermal regulation, right? When that's not enough. So remember this, I showed you this just a minute ago. I basically used these thermal points to create different treatments, right? So I've got a cold stress treatment that's just above their minimum, a heat stress that's between their behavioral maximum and that physiological maximum, and then a control condition. And then in generating all this data, I was able to determine that a 1-hour exposure should be sufficient to get the information that I wanted. I created slightly different heat and cold chambers that were able to keep them at those temperatures for sustained at those body temperatures for an hour. And then I took tissues, did RNA-seq analysis, looked at differential expression and something called gene set enrichment. We're not going to go over the details on this one. I actually just a few months ago in May presented on this chapter, the data on that in this room. And if you're interested in finding out the details, you can go to our IB YouTube site and check it out. You can also see some other cool stuff from other researchers in our unit in there as well. But we're going to jump to the conclusions on this. So really the big picture was that there was definitely a robust transcriptional response to the acute thermal stress. And you can see that here just really briefly on this heat map, where you've got each one of these represents a different comparison of tissue from a treatment and a control, where the red stuff equals there's more transcript copies in that treatment and the blue is less comparatively. Don't worry too much about it, but just there was a difference there. We were able to actually see what was going on. And then notably that transcriptional response was similar between the heat and the cold condition. So they're using the same mechanisms in place to deal with heat stress and cold stress, which I thought was interesting. But also more bigger picture, we saw that there was a big trade-off between surviving the conditions and doing normal activities or normal things that were needed, like reproduction, growth, cellular development, things like that. And so there's a trade-off with these extreme stress conditions and just normal survival. Okay, that was a lot for spring. We're going to jump into a shorter one here for summer, where I was looking at the impacts of climate change on reproduction. And so I looked specifically at an ecologically relevant increase in ambient air temperature on gestation. Okay, set up for this. In the field, we went and collected 50 female snakes that were like moderately sized. These are, we didn't take the biggest or the smallest, but we took fully developed ones that were kind of in the same range. We brought them back to the field station where we did controlled mating trials, where we watched what was going on and made sure that they were pulled out as soon as a successful mating occurred, and ensured that no male contributed to 2 clutches and no female mated with more than 1 male. So it was a single set of genes from each contributor for each clutch that came out of this. We brought them back to the lab here at OSU and systematically separated them into 2 different groups of 25 so that we had an even size distribution between the groups, because body size can play a role on reproductive outcomes. We housed them over the summer in our environmental chambers, where we had one condition was basically modeling current conditions for that region. And then the other one just had a simple 5-degree increase in ambient temperature. So across the board, 5 degrees warmer at night, 5 degrees warmer a day. Otherwise, everything was the same. They had the same food, same access to food and water, same photo period, all of that. And so it was just that ambient temperature difference between them. Then we tracked every time they gave birth, we tracked the litters, where we recorded the date of the birth, the size of the litter, and then the mass and what's length, SVL, snout vent length of each of the babies, so we could find out information on them. And we got really cool results, and we're going to just march through these real quick here. So first and foremost, there was a giant difference, very significant difference between gestation length. So there was hugely reduced in the warmer conditions. So real quick, blue is the kind of normal current conditions, and red represents that 5-degree warmer condition set. And we see that not only did the warmer conditions, did they give birth sooner, but they both started and finished giving birth before the other group even started. So a giant difference in gestation length. I think there was on average a 20-day gap, 20-day difference between when they gave birth. When we looked at the neonates themselves, the offspring, we saw that they were significantly bigger in general. So we saw higher mass and longer individuals for the hot treatment as well. And when we paired that data into a simple BCI where we're trying to track mass and SVL for the same individuals and then compare them across, it also maintained significance. And so we had much larger offspring in the warm condition. There was no difference in litter size. So the average number of individuals born was the same between both conditions. And there was no difference in the percent that gave or proportion that gave birth, right? So out of those that were mated about the same amount were successfully gave birth at the end. So what we really can conclude from this is that there really looks like there's the potential for a huge benefit to both the mother and the neonates from a 5-degree warming during summer. We see that dramatic decreased gestation duration has the potential to dramatically improve maternal recovery from giving birth because of the fact that foraging, that feeding time and gestation overlap. They're actually not very good at feeding while they're pregnant, while they're gravid. And so having 20 additional days in that short summer period to eat could very quickly have them rebuild stores or reserves used for the offspring. Additionally, when they're gravid, they're not very good at getting away from predators, and they do more risky behavior because they need to go warm up more. So it could improve just general survival of the mothers as there's less risky time for predation. And then with the babies were across the board bigger, and it's been shown over and over again that with newborn snakes, bigger is better, both for short-term survival and long-term survival. Additionally, current kind of worst case projections for warming by the end of the century put it at about 5.3 degrees increase, which is very close to what I looked at here. And so likely, even if in worst case scenario, this increase in temperature is not likely to detrimentally impact this specific aspect of their biology. It could actually be beneficial for them in this one specific way. So a couple further questions with this. So where is the limit to this benefit? It's been shown often that you can increase gestation temperatures and see a benefit to it, but at some point there's a cost. You get warm past a certain degree, there's going to start being detrimental effects that start hurting both the mother and the offspring. Clearly, we didn't find that limit, so it'd be interesting to see where that limit is. And then also tracking some long-term costs and benefits for warmer gestation temperatures on both the offspring and the mother could also be interesting to know how that kind of has a long-term effect. And then again, ecological implications. So while the picture is way more complex than just a small thing we looked at, if a warming environment does increase offspring survival and parental feeding time during summer, what kind of implications is that going to have on the ecology around them, right? Their prey populations of frogs, are they going to crash those populations? We don't know. So this would be really interesting to look at further as well. Okay, that was summer. We're going to jump on into the longest part and the funnest part, the long winter brumation period, where I am really interested in looking at kind of how they are dealing with extreme prolonged cold and extreme prolonged starvation. Yes. So I set up an experiment where I wanted to look at, we're back to that transcriptional element. I'm looking at their transcriptional patterns during brumation, right? And so I went and collected 80 male snakes in the wild, or in the field, brought them back to the lab, and we artificially brumated them in the lab for 36 weeks. So we have artificial chambers where we can do that. I modeled this off of previous research that had temperatures that were collected on individuals in the wild, so actually in their den sites during the winter, and I was able to match it really, really well for doing an artificial brumation. At each of 5 time points throughout brumation, I randomly sampled 5 individuals and collected tissues from them. And so those 5 time points are pre-brumation. So we still have a day and night light cycle at that point, and we have normal temperatures, normal activity temperatures. Then as it cools down, we have early brumation, where now we have total darkness. There's no day cycle at all, and temperatures have dropped dramatically, but aren't at the low point. We have mid-brumation, which is probably the longest sustained period during brumation, and it has the lowest temperatures, again, still dark. And then late brumation, as temperatures are rising, but it hasn't gotten back to day and night cycle. And post-arousal, we are back to normal activity temperatures, and we have a day and night light cycle again. I collected tissues and then did sequencing. And in this case, I actually only did sequencing for liver and testis, so I haven't done all 4 of them yet. I just did those 2 for this, for what we're going to talk about. And then I did a time series differential expression analysis. I did some gene enrichment, and I did a gene network expression, kind of several analyses there. And we're not going to talk about these ones for this. So first, we should kind of lay out what the results look like. You're going to see a lot of figures that look like this, and we're going to just kind of talk through it so it makes some sense. So on the x-axis across the bottom, we have time in weeks, with 0 being that pre-brumation, and the one that actually doesn't have a number, but that last tick mark is the 36-week mark, and that is the post-arousal point. For the y-axis, these values are, it's called the expression value on there. And what they are is they're normalized expression values. So there's like transcript counts, but they've been normalized across samples so that they actually can be compared between figures. So this time series expression analysis has 2 basic tests that it does in this process, like thresholds it has to go through. And the first one will be that it tests each transcript for significance between time points, right? So these are the different time points, and it's looking at individual transcripts to see how that works. This one right here, the ATGL, don't worry about it yet, but this is adipose triglyceride lipase. We're going to see more of this later. This becomes a big player in the information that I found. But when we talk about the significance, this figure here has liver in green and testis here in red. And how this would have analyzed this if these were done separately is that liver would have had a significant change between time points and testis would not. It was kind of a flat line. So that's kind of the basics of how that significance looks. And then we have this solid line and dotted line. Those actually mean two different things. So that solid line is kind of just connecting the average expression value between time points. So it's not super informative between those, time points, but the dotted line is going to be a regression fit. And so if we were to say, try and establish what we would expect the expression levels to be at 20 weeks, we would actually want to use the dotted line to estimate that, not the solid line. Okay, so those are the figures. So that's the first step, is it goes and looks at each transcript or each representation of a gene to see if it was significant across time. And then it tries to cluster those based on shared expression value. So this is most of what we're going to see here. Well, we're going to see both these, but this figure here has a cluster, and it's 850 different genes or transcripts that are represented in it that had this same basic expression pattern. And we'll use that to find some more information later. But so this is the step for the time series. It does individual transcripts and then clusters them by similar expression pattern. Okay, so I did this analysis for both liver and testis independently, so I could find out what was going specifically on within them. And then I did a combined analysis where I put them both together, so it clustered them within both tissues together. So all the ones that have two sets of two colors lines are from the combined analysis. Most of the actual work that I based my research on was from the individuals, but they don't show you as much as the combined does. So I'm just depicting these here. All right, there's a lot. I did 18 clusters for each of those three analyses with a total of 54 clusters that I fully looked at in some other ways later. So there's a lot of different moving parts going on here. Because there's so many of these and so much going on, I actually wanted to simplify it a little bit further to help it make meaning. And so I'm mainly interested in the starvation response and this cold response here. So I tried to think about ways that might be informative from the expression patterns, and kind of came up with a generalized expression pattern that would be representative of those. So the two options here for the starvation response, the basic idea is that they don't eat during this entire time period, and they also don't eat when they come out. So whatever is happening in response to starvation isn't alleviated once they get to that post-arousal part. So if it has a change that is sustained, either sustained downregulation or upregulation, I was determining that is likely a response to starvation, right? Because it's not alleviated post-arousal. With the opposite being the cold response, where there is a change, but when they emerge at the end, that post-arousal point, temperatures return to normal, we should see some kind of recovery, right? So those are kind of the two basic ways that I looked at this. Here's examples from actual clusters on kind of what those really look like, not kind of the cartoons I made of them. And so there were many, many that fit these patterns. And so I was able to find several clusters for each of those different patterns and look at the individual transcripts or representations of genes. They had hundreds to thousands for each one of those. And so I was able to go kind of dig through all those and look at them. I further did gene ontology enrichment, where I'm looking for similar types of genes that are expressed together. And I use the clusters to inform this. Basic idea, I would take one of the sets of 18. Like I said, there was 18 for each of the three analyses. I would take one set of 18, I would pull a cluster out and compare it to the rest to see if that one cluster was enriched for any process compared to all of them. I did that for every single one of the 54 clusters. There was a lot of information, a lot going on. When we kind of put that information back into these kind of bigger categories that I made, we found a lot of stuff. There was some interesting things, a lot of it trade-offs that we're seeing where they're turning down metabolism, they're turning down other processes like that, reproduction, all that. Turning up things involved with transcriptional control. So they're really playing a high role in controlling what they're doing. But I didn't think this was super interesting. This is very, very high level. It's very good information, but it's not where I wanted to go with it, not where I wanted to stop. So what I did is I looked through all of that first step, those significant genes, and I tried to figure out, go dig in the literature and find out what those were associated with, what processes those were part of at kind of that gene by gene level, and then build gene networks based off of what was known. And so looking at how all the different genes that were significant that I had evidence for, or transcripts, and see how they tied together and what kind of bigger pictures were made out of those. And so once I kind of put them together, I then went and mapped on the expression patterns and made this giant figure. I spent about a month doing not just this one, but all of these. that was all I did full time, was working on these gene networks and figuring out how the expression patterns work within it. I found some really, really cool stuff. In fact, I was able to build these for all of this stuff, plus a little bit more, that have a lot of detail and tell us a lot about what's going on. Obviously, we're not going to be able to talk about all of these. There's a ton of stuff I'm already talking about. We're going to pick with lipid metabolism because I think it's the most interesting and it's the one I like the most. And we're going to dig into that one and what I found with that. So remember the kind of 3 basic phases that they work with throughout the year. And this will help us understand why this lipid metabolism is important. So during these 3 different phases, we have summer feeding where they get to eat, right, for about 3 months, and that's it. The rest of the year, they don't get to eat. So 2/3 of their life, no food consumption. Somehow they have to utilize or deal with the energy only gathered in the summer for the rest of the year. So that's pretty interesting to figure out how they do that. When you look in the literature, most vertebrates, they use their main lipid fat reserves, these fat bodies called adipocytes, during the long dormancy period if they have it, right? So this is like the standard way, the very high energy reserves. It's a standard way that you're going to store and use energy when you don't have food available. But it turns out for our group and for other members of Sertalis, since about 1985, there's been some studies that come out that say that they're not using those primary fat reserves, that those are not used during this dormancy period. And a lot of people have cited this paper from 85 that has talked about that and found supporting evidence, other things that suggest that as well. So needless to say, I was pretty surprised when I found this guy here, that adipose triglyceride lipase gene, totally upregulated during brumation, right? The whole purpose of this gene is to mobilize stored fats. So if they're not using the fats during brumation, then why are they mobilizing the fats it didn't make sense. So I dug into it and I built this gene network trying to understand what was going on with it. And so once and specifically, this is in the liver. That's a pretty notable thing. So these are hepatocytes. These are liver cells where all of this is occurring. So I mapped on some of the expression patterns here. There's a lot more I'm not showing that I looked at that gave me even more information, but I simplified it to kind of the main players so we can talk about kind of the basic idea of what's going on here. And we're just going to march through this real quick and get some meaning out of it and then talk about kind of the bigger picture. So we'll start here. This is one of the clusters. All the rest of those are transcript-specific expression patterns, but this is one of the clusters from the combined analysis. And for this cluster, it had almost 1,000 genes in it, and it was almost entirely enriched for metabolic processes. So carbohydrate metabolism, lipid metabolism, all these different things. And so I'm considering this moving forward as kind of being the baseline for their overall drop in metabolic rate during brumation. And we see that it was a relative decrease of about 70%, which is in the ballpark of what's been found in other studies looking at O2 consumption and metabolic rates as they get cold, right, of about 70 to 75%. So this is kind of our baseline. And if things have a different pattern than this, then something's going on. So we go back to that HEGL over here, and we see that obviously very different expression pattern than what's here, where it's turned on during brumation and then off again at the end. And I found other genes that were very highly associated with it that had almost identical expression patterns. And so these are co-expression ones. Basically, they all work together to kind of serve the same purpose of mobilizing and using these lipids. So we see that there's a lot of different genes supporting this idea of lipid mobilization occurring during this time period. It's notable that if it's happening within the liver cells, that they are not using the adipocytes, those fat body storage things I was talking about earlier. What they are using is these things called lipid droplets, which are much smaller, contain a lot less energy, but they are the lipid storage sites within the liver themselves. So we see that being mobilized. Then we see more transcripts showing similar expression patterns or upregulation during brumation that involve bringing those now freed up mobilized lipids into the mitochondria where they can be used in beta oxidation to provide energy, right? So now we're seeing some energy use from the lipids during this time period. This one surprised me and took me a while to get my head wrapped around until I found other stuff, continuing to build the network out. But we also see that there's lipid synthesis occurring as well. Two key genes that work together to do the same process to generate new lipids in a very energetically costly process. And so it looks like they're doing that as well during brumation. And then it made a little more sense once I found this, where it looks like they are cycling these lipids throughout, where they're sending out lipids into the system and then bringing back kind of the precursors to recharge them and send them back out again. And we see the one on the right is used for importing LDLs. And that has that similar expression pattern to the lipid mobilization genes. And then this one, the ApoB100, is essential for exporting lipids. And we see while it does have a decrease, it's lower of a decrease than that overall metabolism. We see about a 40% decrease compared to that 70% for general metabolism. So while it's still less, it seems to be maintained a little bit more than most metabolic processes. Okay. There's a lot going on there. But the basic idea is what I'm seeing is several lines of evidence within the transcriptional analysis that show that there is definitely hepatic lipid use during brumation. So they're using these lipids in their livers. And we see increased lipid mobilization, increased lipid synthesis. And I didn't show it, but you also see decreased lipid storage, right? So the genes involved with storing these newly synthesized lipids are downregulated during this time period as well. And then we also see evidence of increased lipid transport, both through an increased import and a maintained export. So how does this stack up to the previous work? Well, like I said, it had already been pretty well established that they weren't using lipids during brumation. However, most of this was based off of those fat bodies, right? Those larger ones that weren't inside the liver called the adipocytes. And it was showing that those don't actually change size over time during brumation. If they did look at more fat types other than those fat bodies, often they just homogenized or mixed all of them up and you couldn't tell where the source was that they came from. And further, that main 1985 paper that most of this was based off of or stems from, they actually pooled sexes when they looked at these. So they concluded males and females in the same analysis or the same data set. More recent work has really shown that there is sexual dimorphism in lipid use, especially during this time period. So that isn't really good that they were pooled. We likely lost some information by pooling that. Additionally, while there was no evidence that the adipocytes were used during normal brumation, when they tried doing dormancy at a higher temperature, they did see a significant reduction in that. And so there does seem to be some temperature dependence on whether or not they're using those normal fat bodies. So the basic summary from what's known is that they're not utilizing their primary energy reserves during brumation under normal conditions. Luckily, I was able to find a very recent, or I guess it's 2019 paper that looked at something very similar to what I found. They set out to look at it. And so they use this Chinese soft-shelled turtle. It's a different setup, but it's a very similar concept in how they do their winter dormancy. And they ended up finding very similar things to what I found, but they did a lot of work with the more physical side and not the transcriptional side as well. So they saw that there was increased lipid mobilization within those lipid droplets in the liver. And you can see that on the figure there, where during brumation, they have much, much fewer of them. They saw an increase in the free fatty acids. So this is when the lipids get freed from those lipid droplets. The liver had more free ones floating around. And then additionally, they looked at a lot of the same genes that I looked at, because again, those are the major players in this, and found that they were also upregulated during hibernation or brumation, right, during the same time period. They don't have the time series expression stuff, so they're not getting like the fine detail, but we see that there's more of it during hibernation than there was in non. So that kind of backs up some of what I found. It gives some kind of validation to there actual are changes to the lipid content in the liver during that time period. So what does this all mean? So what this shows is a temperature dependent energy substrate switch, where when I map temperature over the expression pattern on this, you see it pretty much mirrors it really, really well. Where when it gets colder, these genes are turned on to free up lipids within the liver. And as it heats back up, they turn back off. And what I think that means is that they get to change their substrate they use depending on environmental conditions. And so during this really long-term winter dormancy, they have things in place to rely on lipids stored in their liver specifically. Even though it's less desirable, it's less energy dense, they still are able to mobilize those and use them during the winter dormancy so that when they come out in the spring and it warms back up, they can switch to their really energy dense, really high energy adipocyte or fat body stores during winter or during spring. So why would they do this? The spring mating season we talked about in the very beginning is like the most energetically costly period of their entire lives. Additionally, they're still not eating during that time. So they have to put out a whole bunch of energy in order to reproduce, right? And so if they can avoid burning through those energy stores during that long dormancy period and maintain them for when they come out, they're primed and ready to go to use all of that to do this courtship behavior and be reproductively successful. So I think that's a really, really cool possible situation going on there where it really explains what we could be seeing here. So I did this for a whole bunch of different networks. Like I said, I made a whole bunch of figures where we tracked expression patterns, and I found some really cool, neat, different stuff that we're not going to talk about today. But it's there. There's a lot of information. I really, really like this time series expression pattern way of looking at this, because I think it gives you a really good insight into a bunch of different things that are happening. But yes, that's for another time. So, with a lot of stuff, really, really fast. But we just marched through the thermal year of these garter snakes. And we can kind of summarize what I found overall in my years of research working with these guys. So first, we see that they have tightly regulated thermal preferences and limitations. And this is consistent across geographic range and time period within the same place. And along with that, that means that they're likely using behavioral adaptations to overcome these environmental changes. We saw that there was strong transcriptional response to thermal stress, and that both heat and cold stress gave very, very similar responses between them. We saw a very clear trade-off between maintaining homeostasis, so staying alive in stressful conditions, and reproduction, cellular repair, growth, and development, so things that are really essential in general. So they had to pick between the two of those, basically. And we saw that in both the behavioral thermal regulation, acute thermal stress response, and that brumation long-term thermal stress. So we kind of see that across the board all throughout the year. They're having to do kind of these trade-offs when things get tough. Interestingly, we found that not all potential impacts of climate change are looking to be directly detrimental for this population. So we saw that warmer summers may improve neonate fitness and may help maternal survival and fitness as well. And then finally, I really thought this was fun that we found these thermally dependent switches during brumation, and there were several aspects of metabolism and stress response that showed this pattern. Again, I didn't get to talk about all of them, but there were a ton of different places where we had a very clear thermally driven change in how they handled things. So for future work, there's a lot more than this, but just basically, so I talked how I used mostly males for the thermal biology, or most of the thermal stuff. It would be very interesting to repeat it with females. They're a little more complex in elements involving that as far as reproductive state and size variation and stuff like that. But it'll be really interesting to see how they stack up and how different they actually are. I would love to do more of these time series transcriptional response stuff. Specifically, I think it'd be really interesting to look at the acute thermal stress, but over time instead of just not stressed versus stressed, and see how they regulate things to deal with that stress. And then there's a ton of stuff that I found in the brumation work that I would like to validate, actually go in and look and find changes to different tissue types or protein abundance and things like that. Okay, so for acknowledgements, there's a lot of you. First and foremost, my family, my wife and kids, thank you so much. This has been a long road. We're almost there. Bob, it's been, especially this last little push has been really, really awesome. I appreciate that. My committee, thank you for bearing with me through COVID and children and all that, challenges involving that. Kind of got my primary mentors outside of that, Lori, Adam, and Aaron. Thank you all. Mason Lab and other grad students I've worked with. Office staff, obviously, we couldn't do any of this without any of you. And like I said, well, wrote in another place, you've probably helped me more than I'm even aware. So that's awesome. Some people at my community college were a big impact on me, and some others that helped me build the confidence to get where I've gotten today. And then lastly, all of you here, all my family and friends and colleagues that showed up, I appreciate your support. And one more little thing. So when I went to make my flyer and make this presentation, I looked everywhere for a picture of me doing my science. It doesn't exist. I have not a single picture of me doing anything that's related to my work. But what I did find was a whole bunch of pictures of my hands holding different animals. So I thought that was kind of funny. This isn't even all of them. But what I also saw was that I seem to be passing it along too. So that's cool. All right. Thank you. Time for questions.
So Yep, thanks, Dave, for that talk. Excuse me, the tradition or whatever the custom here is for anyone in the audience to ask questions except for his graduate committee, because we're going to take him after this and grill him like a burger. We'll have our chance to do that. So go ahead, Ricardo, you got some thing?
Yeah, the first question is for physiological thermal limit testing, are there any long-term adverse effects during testing these limits? Did these animals get euthanized following experimentation, or are they able to recover?
That's a great question. So in doing those, we actually had so they recovered quickly. As soon as they reached those points, we put them back into normal thermal conditions, and they rapidly changed. We're talking the entire experiment for the heat stress took like 8 minutes. So it's a very quick change in temperature. We returned them to normal conditions, and they were monitored for, I think, 2 days before we let them go back into the wild. So almost all of them went back into the wild. There was a single one out of all of the things that I did that didn't make it. And it was during the heat stress one. And it actually had a very uncharacteristically fast response where it didn't recover. It reached its threshold way lower temperature than everybody else and didn't recover. So that was the only one we lost. Everybody else was completely fine and got returned to the wild afterwards. Yeah.
Oh, nice job.
Thank you.
So with climate change and like expected maybe increases of like warmer winters, do you think that will influence how they are using things like lipid stores, or if they have to go into these fat stores earlier than they usually do after permission?
Yeah, that's a really good question. So I thought a lot about this one, and what I think the real basic answer to this is that I don't think it's going to make a big difference because of how cold the winters are. So maybe at the edges, right, the transitions might get closer together in time, but I think throughout the duration of the winters, you're not going to go from -16 to +5. If that occurs, we're going to have some serious problems, right? And so because of how cold it is and where they sit at, I don't think they're likely to be impacted during that time. Yeah. Yeah.
So it might be a no, and you may have sort of alluded to it, but so based on the 1985 paper, they pulled males and females, correct? So it kind of changed the outcomes and what they saw with lipid usage. So if you're seeing in just doing males that there is some sort of lipid usage, although it's coming specifically from the liver, so that might change some of how that is quantified. But what could you speculate is happening then with females if that's changing the results by adding females, and like what could their storage be that they're using, or is it different because it's coming from the liver?
So some of the big differences, the females have a way higher percentage of fat bodies during all of it. They also need to produce eggs when they come out, and so, or not eggs, but egg yolks for the offspring. And so there's just a lot more going on there. I don't know. It would be interesting. I don't have a great answer for that. I know I would expect it to be different. I think that it's still possible that they could be doing something like this, but it was just such a widely different thing that's been shown already with them, but I'm not sure exactly what those differences would be all the way. Yeah.
For the lipid metabolism analysis, the testis was simply the negative control in comparison to the liver transcriptomics, beautiful Western blot at the ATGL protein. No, that's the question.
So I wouldn't, the testis wasn't really the control. I guess you can see the difference in expression, but because it's comparing between time points, right? There's not like a control not being in hibernation or cremation thing. It's more or less, was there a change between points? And so I think that's kind of more where that sits. I may have missed the question in that.
Next question says, you compare the active DVD to other snakes in their genus and found lower values and attributed that to courtship and mating, but is the range different such that there might actually be adaptation to lower temps in the species?
So I guess maybe that wasn't totally clear. So for this species, during other times of the year, they actually do tend to have that more preferred body temperature closer to 30 degrees. And it's been found within the species that during the courtship time period, they don't regulate it as heavily. And so it is often lower. So it's not, I don't think that that is their year-round preference. I think that that is just a result of being in that courtship behavior.
Again, great talk too. I've seen it like 3 different times. And I've probably asked this question to you already once before. But I was really curious about for your, when you increased the temperature and you saw changes in growth rate and stuff, you really only compared the neonates and the mothers. Did you, or have you thought about like what happens as they keep growing? Like, so they grow bigger when they're young. Does that actually carry over into adulthood? Is that like a trait that maintains as like, you know, potential positive, or is there some other trade-off that actually, you know, with global warming, climate change, does that actually affect all of that cycling stuff down the line?
Yeah, so I do think it's really interesting looking at the long-term effects. And let me clarify, are you asking a sustained increase in temperature, or are you asking just now that they've been born bigger and sooner, do they have a sustained benefit?
Yeah, the second part.
Okay. So the sustained benefit, the sustained benefit actually has been shown in general, where they've just looked at like offspring size and how that's affects them. And so very specifically, it was very clearly shown that it gets them through that first winter dormancy period better. It makes them much more able to get through that. And then they actually had some other papers out that showed that there was a better long-term survival, that they made it to reproductive maturity, or not faster, but more successfully. And so there likely are long-term benefits from that. Yeah.
So follow up, when are you designing the selection experiment to do all
Yeah,
that?
right. Well, see, they it takes 7 years for them to get about reproductively mature. So, you know, maybe I spend the rest of my life working on that one. Yeah. So, yeah.
So my question is about the future. You did all this science, and it's unbelievable, quite frankly, David. What are the questions you have from doing this you want to pursue next from this?
I tried to get kind of an evolution element into this, and it just didn't work out very well with the samples that I had. I actually went through the whole process and got to data analysis, and it just didn't work. And so I'm not even necessarily in the snakes. I'm still more interested in kind of the impacts of thermal changes and evolution and how things will adapt at that level to things. And I would really like to explore that more. I think that that would be really interesting. And of course, as I keep saying it, the time series expression data, tying that into things I want to do more of as well. I just really think it's got a high potential for finding cool stuff. So, yeah.
So you mentioned that the same genes respond to extreme cold and heat. Does this suggest that the response is more about damage control and or direction of change in expression, or just the opposite?
I would agree that, yeah, it's likely that it's a response to maintaining protein homeostasis, a mouthful of words, but when temperatures get too hot or too cold, your proteins will not function properly. And so you have things like heat stress proteins, which is kind of the primary response that was similar for hot and cold, that their role is to help make those proteins function properly again. And so it does make sense that they would have a very similar response in both the heat and cold stress environments.
To follow up for that, do you think there might be differences in how efficiently they can use adiposate fats versus liver fats, and that this might contribute to when they use the different stores? For example, if the liver pathways can produce free fatty acids as quickly as adipocytes, they use those when energy needs are quite low or slow. Finally, larger Lego response. It's a long question.
Yeah, I'm trying to wrap my head around it. So I think what I got from that is that it might be, we're talking like a location difference, and maybe that's explaining it, that it's harder to get the fats to the liver from the adipocytes versus the ones that are local. Does that sound okay? And that actually makes sense, especially with it being really cold. It's harder to transport things and move things around. So that could be part of the reason why. Either way, the end result still is there, that when they warm up, they have the stronger or the larger energy reserves available for reproduction. So I'm not super certain on the exact mechanism. That's what I want to look more into. But I think the evolutionary consequence makes total sense that this is a thing that's real. And so I think it would be interesting to kind of further look into that. Yeah.
So I know in some organisms that, so they had like ground adipose tissue, which is more found in juveniles, right? To have those important fat storages. Do you think there's a similar thing in garter snakes where the juveniles have a specific, adipose storage compared to adults?
So I didn't look at juveniles for this part, so I'm not sure. There's a lot that we don't know about what the juveniles do. They don't come back to the dens during winter, so no one knows how they survive winters until they mature enough to come back to the dens. So it's a whole thing that could be looked at, but they get born at the summer feeding grounds, and then we don't see them again until they're maybe not fully reproductive age, but close, several years before they come back. Yeah. Yeah.
I am curious about the leafage used in the winter. I will hypothesize that metabolism decreases significantly in the winter. I might have missed this, but do you have a sense of how these both snakes compare to snakes in the summer in terms of this pathway?
So I don't have any expression data for the summer at all, no. But I did show kind of the general overall metabolic drop, where the one cluster that was enriched for right around 1,000 different genes showed about a 70% decrease, which matched what's been published in the literature for their overall metabolic drop at those same temperatures. So I do think that is kind of the baseline we were looking at. But again, all the things that I was talking about with lipid use showed that instead of dropping at all, they were being turned on, right? They were increasing during just the cold periods and were not on during normal thermal conditions.
All right, I think, yeah.
I have more questions here.
I have more questions. I'm going to have to intervene and just say, okay, so thank the people online for having those questions. I'm going to say, well, they'll have to
reach out. Yeah.
David afterwards. So one last thing before we go. So normally it's our custom, again, well, we have to, we David's graduate committee, we're taking real emphasis in private. And then normally friends and so forth wait around in the lab, and then we either pop the champagne or commiserate with the I'm really building this up right. Not many of them matter. But given the fact that we don't live in Corley anymore, so our labs are literally open to the whole environment. So our whole department has been moved off campus, which would be great when we come back. Anyway, the point is, so rather than celebrate the near term right after, we're going to celebrate this evening. I don't know what it'd be, 4:30, 5 o'clock, something like that. We're going to go to the block 15 in Southtown, not the downtown, the Southtown.
A tap room.
And rumor has it that his advisor is buying all the beer.
That's a good rumor.
You like that? Yeah, he's graduate students. Yeah, but there's all graded there. Yeah. Okay. Yeah. Just the grads. All right. Well, let's thank David for that.
Thank you for coming.
And the leading members, I guess, really was our play.